Hey everyone, the first data are available, the second lane is running. And so in order for us to look at our data, I thought to briefly record a tutorial of how I analyzed Kelly's data. The idea, of course, of this tutorial is I cannot get everyone up to speed. You all have different backgrounds, but I think with the help of a bioinformatician who might set up Jupyter Notebook, as well as this notebook for you to edit, you might be able to get a quick handle on your data. So the first thing, what is Jupyter Notebooks? It's a really cool tool that allows you to code in your environment. So if this all doesn't make sense yet, you basically have these windows for your code, and then it tells you what it's doing. Jupyter can handle R, it can handle Python and Bash, and most of my analysis is done in Python and Bash. So what you would need, you need Jupyter installed. If you, like me, use Windows, you use PuTTY to get Jupyter connected to a server. Aside from Jupyter Notebook, you need a couple of packages installed. You can find all these packages on Bioconda. And on top of that, you will need to have installed, of course, Homer. We are a little bit biased here. So diving in, I will share this notebook with you as well as a bunch of the files, and you can get started. A quick heads up is, if you run into problems with your own data, maybe download for something simple with a strong phenotype like the KLA data from the 2019 paper, or contact me and I can give you some boring K562 cell line data. But it's always good to first start your analysis to get confidence with something that works. Okay, so to start off, I had to import all these packages and generate my directory, which is basically, I just called it 2021 September mouse lungs. I generate 4 directories called raw, analysis, tech during QC, and quickly check that that happened, and copy over my data from the Dmux folder. And generally, I like to always keep the raw data with my analysis. So in case anything gets published and some people ask me to upload things to Geo, I know exactly which files they are as well as name them. So, okay, all the files are here and you see already what happened. This is because this is a bit out of order. So let's ignore that for now. And when I first looked at the data, they were not exactly named the way that I needed for my script to work. You might remember we had our initials. Well, that's what this little piece, it's just a little loop, says that everything that's called .gc, remove the KC mouse and call it mouse to basically get rid of Kelly's initial. And then it was my mistake that I didn't communicate it, that I need the R1 for the replicate behind the method, as well as the lower score versus the dash. But basically, this is just a quick loop to just go through all samples and rename this into this. With that, all the samples were named in a way that my script likes to do it. And we were able to generate tech directories. For that, we first need to map the data. One cool thing about Jupyter is this is all written in Bash. So you can just use this %% bash to now just write in Bash, although this is a Python environment. And because I'm curious, I always add the time. So depending on your computer, this will take a while. The cool thing is everything that's in Bash, you can also just type down on your command line, no problem. Okay, what I'm doing here, we actually trim our adapters. And this is very critical because you remember the small RNAs are between, let's say, 15 to 60 nucleotides, but we sequence 84. So if we don't trim off the adapter and we don't do a good job with that, our data actually won't map. I love to use Homer tools, and you can also just use the traditional Homer trim as on the website. But there's this neat little batch parallel.pl, which allows us to trim across multiple. So it's just a multi-threading version of the trimming. And batch parallel basically just runs this command that's in rabbit ears in batch parallel. Otherwise, you can also run Homer tools, trim up to 3 mismatches. This is the adapter sequence. Sorry, -3, minimum up to 2 mismatches, minimum length 40, 20, and go through all the fast Q files. This is something which might be a bit specific to me. I like to automatically zip up the files just to reduce the space and then map it using star. So some of you might know that star is a splice aware aligner, but we don't need splice awareness for nascent data such as csRNA-seq or maybe even GrowSeq. The reason though I use star in this analysis, because to the best of my knowledge, we will also generate RNA-seq data for this project. And if you have RNA-seq data, you will need to use star. And I'm personally not a fan of combining directories or data that were mapped with different alignment tools. So you basically cater to the lowest means. In this case, it's RNA-seq. So we're going to use star for mapping for star, because this block here probably runs 2 to 3 hours, depending on your data. Again, a quick loop, which is just for all the trimmed.gc, and because it's trimmed, you need to have this ccut file. This can happen a few times because I'm actually still analyzing data in the background on my other computer. So so I run star. This is the number of threads. The genome directory, you need to have a genome, but that's pretty straightforward. Everybody can help you to install that. And then the read-in file is basically I, which are all the GC files. So these are basically standard star parameters. So it doesn't time out, and it's going to loop through all the directories and map them hopefully to the genome. So this is basic alignment, and I hope some of you have done it before, and if not, copy paste it. I hope it works. For the next thing is I want to make tag directories, and to do so in my environment, I like to use these sample info file. What this file basically just does, it's a list between the SAM files that were generated by star and how I want my tag directories to be called. So I look for all the files that have .sam, which are the output files of star, load that and edit this a little bit around, and then save this as my sample input file. Next, I'm gonna run the batch make tag directories.pl. So here is some bad news for all of you. When you see these longer links, this script is not yet part of the current Homer version. So to get that script, you can either contact me or Chris Benner, and he will share that with you. The good news though, however, is if you have less samples, you can just run the, as on the Homer website, the make tag directories.pl. So this is again just a batch version because we started to have too many samples, particularly during the COVID studies. If you have hundreds, 200 tag directories, then basically it is good to have. Okay. Apologize for that. Where were we? Here, sorry. So this is basically 50 CPUs, genome mouse in this case. The important thing to point out is to use the check GC function. That is, so you have some files saved that you need for QC later. If you want to analyze nucleotide frequency plots and so forth. And if you use this script, the -r is a nice function because it basically combines the two tag directories if you have replicates, or if you have 3 replicates, that's why we use the -r. So if that's too much and already confusing, that's fine. Just go ahead. For those of you who are familiar with that, there are different ways of analyzing data. For example, we can pool our samples or we can use them as individuals. My personal preference is always for calling features, let's say peaks, I like to combine my tag directories. And so that's why this -r function, it generates me both a combined tag directory profile as well as the unique ones. Let's say you have 3 replicates, you would get replicate 1, 2, and 3, and the combo one if you have the -r function. The -r function may not be available at make tag directories.pl, so the non-batched version, but here you just use make tag directory.pl and then add the 3 tag directories and the new tag directories, and then the 3 will be combined. So for calling features such as peaks, I use the combo. For annotating features, or stuff like the e-seq for differential gene expression, I use the individual tag directories, so each replicate individually. Cool. Okay, so now we have our tag directories. The first thing, oh, that starts to be annoying, is we want to load this length file, because one key QC of csRNA-seq is how did the read length that we analyze in our libraries change? Because remember, we want to deplete for the 21 nucleotide microRNAs. My coding skills are not the nicest, so I manually have to give one of the length files from the file. So you're just going to use your ls command and then basically look for it. And then I concatenate all the files that have this cs input-r. Right, this is all that this little code does, and it spits out this little nice graph to me that shows, wow, beautiful. Kelly did an amazing job. You see, these are the input libraries. You have this huge peak around 21 to 23 nucleotides, which are the microRNAs. They're very little adapted dimers. And so this is basically how you expect, particularly in mouse, the input RNA to look like, right? Remember, we didn't see anything on a gel, but boom, here it is. So now we actually do the same plot for everything, but it's not an F. So basically, if it's not these ones, then these are the csRNA-seq libraries. And you can see here, they look quite a little bit different, right? You don't have this strong peak at 21 anymore, so that got drastically reduced. Therefore, you have an enrichment of these areas, and these are actually our csRNAs. So that's already a very nice indication that this worked really well. And of course, you can also plot them all together by just basically putting here select something, a string that is common to all of them. Let's say in Kelly's, it would be KC or the ES, which is the sample number. So that's already good news. And so the next thing we want to look at is the mapping stats. And to do so, we look at the raw data, and in the mapped file, there is this .final.out file. So the problem with my script is that I like to gzip the files up, which actually calls the final file gzip log. But Chris's little script is looking for log.final.out. So to make these files recognizable to the script that's part of the HUMA pipeline, we'll just have to rename it, basically make it log.final.out. Once it's renamed, we can basically just use the get HUMA QC stats.perl script. As with the files, the nice thing is once you have the sample input file, this will tell you all the tag directory names. So it will find the length files there and make the stats. So this stat will be printed out in your QC directory. So all QC related ones like this picture. And so I save in a QC one. But I also loaded, rename it, and delete a bunch of stuff, as you see here, that I think is not necessarily important for csRNA-seq. Remember that script works with all the different measures, and if you have RNA-seq or ChIP-seq, you might want to keep some of those different ones. But in my case, I'm only really interested in total reads, adapted dimers, alignment, and so forth. And for simplicity, this mapping stats, which is going to get printed out, just end it here so you see what's in there. So these are the library names, the total reads that we got, which is a bit between 15 million for csRNA-seq and then a little bit less for input. Very good job here by Kelly. Pretty low adaptive dimers, which also led to nice alignment tools. The thing to perhaps comment as well on the stats that it's worth a look or two is this total positions in analysis. So you see already that the total positions per input are a lot lower than for csRNA-seq. This is because these microRNAs and highly abundant small RNAs just suck up the reads. So you can sequence them pretty deep, but you rarely get more than 200, 220,000 positions, at least in mice. While in csRNA-seq, you get a bunch of total positions, and here Kelly is actually rocking it with 3.5 million, which is pretty awesome. You want to have about 1.5 million total positions to start to be confident in the analysis. 2 million is nice, so this is all working pretty nice. The other thing to look at briefly is the average read length. To see if you have good csRNA-seq libraries, this number should be in the order of 35, 34. And for your input libraries, it's 23, 24. And so this is also why, if you remember, the calculated input length for csRNA-seq and input libraries varies. csRNA-seq is about 13, 14 nucleotides longer. So together, this table gives you already two key QC numbers, which is the length as well as the total numbers in the precision. But whatever you do, the most important thing is always to spot check your data on the browser. I personally like to make my browsers sessions different or individually for input and csRNA-seq. And this is just because of auto-normalization. So what this little thing just does, it goes through and selects all the ones that are called csRNA-seq. This is the variable, and you see these are all the tag directories that have csRNA-seq in them. And then I generate the hub file, this make multiwic.pl, give it a name. This is the genome. .tss is the parameter that I highly recommend you to use because we actually want single nucleotide resolution. I like the gradient, it looks fun. And then as tag directories, as again, I use that and I generate one for csRNA-seq and one for input. When we look at that, the first line is basically what you will need. So if you go ahead and copy that, you go in the Genome Browser, My Data, Track Hubs, and this is where you would copy it. In that case, I already looked at the data. So let's look at the Genome Browser. And so this is a typical example of csRNA-seq. This is the ribosomal protein 1. I like it because it's extremely strongly expressed in RNA-seq. But when you look at it, you actually see all these start sites here. But if you look at it twice, you might realize, oh, actually, these ones are sort of on the upper strand, the Watson strand. So these start sites go this direction, and the gene is here. And indeed, the gene is a very focused promoter that starts on this CA in mice. And yes, we have this start site that is nicely found in our csRNA-seq samples. But what is actually dominated in this area is this sort of downstream enhancer. And you can see these are unstable start sites that go in a different direction. Another one that's quite often worth to look at is this MALAT1 gene. And this gives you just an idea because MALAT1 has this crazy long axon. And you look it out. And so the question is, when you go across this long axon, do we actually see any significant csRNA-seq reads? And there is some, right? If you look at a scale, it's 12, something like that. And then when you zoom out, there's some MALAT1 enhancer. And here you see that you actually have, particularly in the input, but also in your csRNA-seq, a good signal throughout your samples. So in generally, csRNA-seq should be depleted for the exons. And if you're curious, you can also see, okay, is the gene expressed? And at least in these samples, it doesn't seem to be. Okay, so I actually don't know what these samples are, but another spot check is probably, oh, it's called B1. Yeah. If it's something, yep, so something in there is an immune cell, so it has Q1 expressed. And so a typical thing for Q1 is also that you not only see the start site, but you also see this downstream Q1 enhancer. So look at your favorite spots. Another one, of course, that I personally like is ID1. ID1 is not so interesting from this whole enhancer world, but why ID1 is pretty fascinating, at least to me, is that at least in humans, it has a canonical data box. Let's have a quick look at that. And yeah, TATAADA and then TCA. So this is the data box, this is initiator, and therefore the transcript should start on the A and you nicely become the start site. Okay, why did I spend so much time on that? It's again, it's very, very important to really look at your data in the browser. And I would say if you get from demuxing to browser session, that is already a huge, 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 huge win. And that's all I could do for many, many years. Because then you can experimentally spot check your data and see, okay, is my gene of interest up regulated, down regulated? Are there any connections? How do my data look? Yeah, so that's what I would say, definitely do it. Okay, so this is sort of the first part. The second part now is we want to define our features, which we can call peak finding. And you see that here. Let's just make a directory called peak calling that already existed. And to do the peak calling now, I need to make one list that calls all the csRNA-seq files and all the input files. Because remember, each csRNA-seq peak is called relative over input. We do not have the RNA-seq data yet, but if you have RNA-seq data, you add them in here, and then you make sort of this triplicate. And implementing the RNA-seq data is pretty cool because it actually tells you if there's any RNA-seq within the 150 base pairs of your csRNA-seq peak. And if there's not, it's called unstable. And if there is some, then it's called stable, and it actually calculates the ratio for you. And of course, you know, enhancer RNAs, and these ones tend to be unstable. So this script is a nice little tool to look at stable versus unstable RNA, which I'm a bigger fan of than using promoter proximal versus distal, as these annotations were made on cell type X, but you study cell type Y, and start site commonly change between different start sites, cell types. So that's why we don't have an RNA file. This is the GTF. We work with mice, MN10, and I use an n-tag threshold of 10, which means I want a minimum of 10 tags enriched. Again, this is the latest version of the script that's not yet available. I think what I should just do is get these two scripts. And if you're familiar with how to save these scripts, contact me. If not, maybe ask someone who does some bioinformatic help for you, and then we can share these scripts. This is, again, this file that we just generated. And then again, the mouse genome, in that case, I used MN25, MN10, and a text threshold. Cool, then this finds the peaks for us, which is really nice. And these are now TSRs, so the analysis currently is not done on single nucleotide resolution, but on peaks. Then the first thing we want to do is merge all those peaks and annotate them. And I annotate them twice here, once using raw counts and once using rlog. Rlog is a normalized version. And for example, what we can do with the normalized version is this clustering as done here. I just took everything and had as one, pick up one. This is actually rlog, not RPKM, sorry. And I just split this one here to make this look nicer. And the data start in line 19. So long story short, between 9 and 18, the columns between 0 and 18 is just garbage, which you can't really plot, like which gene it is, the coordinates, and so forth. So here we just want the raw values and generate a cluster map. And as you can see here, also save it in the QC folder. And what you can see nicely here in Kelly samples is two things that are important that you can see. One is that the same names sit next to each other. So that's pretty nice. The other thing is this sample is sort of the outlier. That's the furthest away. And then these form one clustering and these form one clustering. And if you can look at that, I believe this is one strain of mouse called DBA20. And this is C57. I think this is then DBA and this is C57. So basically, even the mouse strains cluster together, and then within the cluster, these are hypoxia, normoxia, normoxia, hypoxia, which basically tells me already that in these lung tissues here, the strains cluster differently. I believe this is a young mouse. These are old mice, but basically then they cluster by age, and then they cluster by treatment. But it's always nice to see that your replicates cluster together. A bit more fancy way of doing it is this PCA. It's pretty straightforward. You can copy that that code here. You transform your data, check, okay, that looks good. Then I quickly check my columns because I need to copy the columns names here into the targets. Need to adjust my colors. These are other color options. And then basically you can go through, and in my case, we use these 10 tag directories using these 10 colors and can do our PCA plot. And that also basically tells us how similar these samples are. And then here, principal component 1 is what really separates these different lung samples in DBA mice from the lung samples in all other mice. But also in the other mice, you can see here, on the bottom, this is C57, and here on the top is DBA. So all I meant to say is our samples nicely cluster, not by normoxia versus hypoxia, but by strain, and then whatever the difference between these DBA mice and these DBA mice is, between 20 and P and D. Right, with analysis, the dirtiest way to just look whether there's something interesting is to use clustering. And one of the scripts that hopefully will be published soon, but it's already we can provide it already, is this do it all dirty thing from Chris. I mean, you might know him. He coded Homer and was a large part of Metascape and Mealop and so many cool programs. So at some point, Chris decided to put it all together, and this is this analyze cluster script. What you basically do is you use your alloc files. You define the peaks. This is mouse. This is the size of the peaks. And then there's a threshold. And usually I use 0.6, but I thought to do 0.4, 0.6, and 0.8 here. So now what is the really cool thing about this script? It looks for clusters, and it looks for motifs enriched in clusters, specifically in these samples. So for example, here, these DBA ones, right? They are highly enriched for this KLF zinc finger versus these others. But then on top of that, the top one GO term is this regulation of lymphocyte activation. So why do I advertise that script to you? It's an easy way to get the top motif per cluster as well as the GO term per cluster. If you wonder, what is a cluster, what I'm talking about, for example, oops, let's go back up. This might be a cluster, or this might be a cluster, or this might be a cluster. And so the neat thing about the script is basically it automatically tries to take your data apart and find interesting trends. For example, this KLF1 cluster is pretty much common to all the C57 ones, right? While we might find, yeah, this is more unique to these older ones, same as this one. And then this cluster here, not much, but it's a little bit more DBA specific. And it's this FLE1-ETS motif and a GO term that's associated with this cluster as well. So this is just a lazy way of trying to see, oh, is there something nice and small? But again, this might also be a little bit complicated because it basically throws all genomic hammers at your data at the same time. And when you do something like that, you want to have different thresholds to see, okay, do we recover the same motifs? Do we recover the same trends? And again, you see here, okay, this is the KLF, which is basically a GC box that is still pretty much here and also in these clusters, which tells me, okay, these ones have really long SP clusters or GC box clusters. Right, as this could be more confusing, I think this is basically for the geeks among you, a script that I highly advertise. But traditionally, what you want to do is DE-seq, right? So let's make a DE-seq directory. And for DE-seq, we actually take our raw data, so not our alloc data. Again, in DE-seq, you want to generate a data frame that just has an index as well as your values. So I just take 0, which is the first line, as an index, and then 19 to 29 because we have 10 samples. So the TSR ID, as well as all these sample names. I usually just like to use dot columns and then copy these into my conditions to make this condition string. Then use the getDiff expression.pl. This is the raw file of all TSS. TSRs would be better, so transcription start regions, it's not transcription start sites. These are the conditions. And then I export the peaks and I give them this all conditions prefix. In this case, I look at all versus all because it's, if you have like a control, you leave that out and make sure that your first 2 samples are a control, then everything is by definition compared to your first samples. But in this case, I really don't know what they're looking at. So sometimes you analyze data and you don't really know what people want. So I just look at all versus all and save that in this all conditions TSV. And what's pretty nifty is it already spits out the number of differentially regulated genes. And you can see, for example, this normoxia versus hypoxia didn't really cause much change. But if we look at, where do we see it, for example? So this is the same condition between these 2 different mouse strains. Then you have about 1,000 to 2,000. So in total about, close to 3,300 TSRs that changed at log 2-fold. So 2-fold FDR 0.05. And these are, of course, promoters and enhancers. So in a traditional way, you might be interested, okay, but which motifs are enriched in these files? So let's do motif finding, generate a file. And oops. Did I do clustering again? Okay, sorry. Did the clustering again. That jumped down. Here we go. Do motif finding. So again, this is our DE-seq. Sorry, that jumped on me again. Make the motif finding and then, come on. Sorry. Oh, this is because I am still doing motif finding. This is the script that's still running on my other computer. I apologize. I should finish that before coming. And that's why it's also messed up. And there are 3 different ways of doing motif finding, right? One is, so you can see the DE-seq. Undo it. The DE-seq spits out these files for me. All right, so DE-seq spits out these files for me. I'm not going to click again. And so one way is, of course, you can just look which motifs are enriched in those up versus those ones that are downregulated. Like, so basically take 1,000 peaks as your foreground, the others as the background, and see which motifs are in my upregulated genes versus the downregulated loci. And then you can also do the other way around, of course, which ones are in the up versus the downregulated. The second way of doing motif finding is you basically do not give a background, and then Homer will GC normalize the background. And this is also an important control because you can imagine that, for example, certain regions of the genome, like promoters, have more GC, and therefore you might find the GC box, so SP1. While then when it's GC normalized, motifs will become, enriched rather than just based on sequence bias. And the third one, which I actually commonly like to use, is I just take all my peaks and then subtract from all the peaks those ones that are enriched, which is just done by this co-bound command, give it a prefix, and then here it's important to use the cobind 0. When you do that, it makes sure that the peaks that have all of them is first. So it basically says, these are now the peaks that are in here, but not in this file here. And then again, you use findgenome.pl. This is the, for example, the ones that are downregulated in this condition, mouse 10. This is the folder that will be generated. And this is my background file, which in this case is all peaks minus these ones. There is, of course, a lot more details in here. Unfortunately, that's where it bugs. But you can see the details here. Size, I take minus 150 to plus 50 to the TSS. You can use minus 200 to plus 50. But this is basically where most of the sequences are. I look at length 8, 10, and 12, and I only look for the top 20 motifs. In this one, it's going to spit out the data in this directory. I personally like to use, oops, does it move? Nope. One more try. This WinSCP software to just download it on my computer. And as I said, one of the reasons this was bugging is because on the other computer, this was still running. But here are two of the files that are here already, which is basically the motifs that are up versus down regulated. And the HOMA files are the novel motif finding. You see, okay, this is GC rich. And when you see there's a lot of these different patterns of GC, you should already be a little bit nervous, and it could also be because you have low numbers. In these cases, it might be better to look at the known motifs. And basically, you see in the known motifs a little bit different. You see there's a lot of difference in ADS motifs. Yeah, basically a lot of ADS, and then it also finds this FXM nuclear receptor. While in those ones that are up here, let's see, so that the novel motifs, MIP, CBTV3, okay, Rung SCADA, another ADS one. But again, we can also use the known motifs. And so these are motifs that are previously identified. But one thing to look at is the p-values are really, really low. And a lot of those motifs are just commonly found in promoters. So this is something in this sample, at least, it's actually good, maybe that it came up. These are not motifs that I would really trust much. As you see, the p-values are extremely low. Another way to look at that is that the background is pretty high. So that did not work too well, and that's okay. And let's see, maybe some other ones are finished by now. And let's just download these ones to have more look-sees at different ones. And so now this is the one versus using the TSR background. Again, pretty low p-values. And you find NF1, NFY, CBT, which are typical promoter elements. So, but at this, see, it's in 0.93% of targets. It's nothing really specifically enriched in here. Same in the known results. Okay, typical co-promoter factors, nothing there. So, no, still loading. I'm gonna pause you for a second. All right, it should be done in a second. And so now this is versus random backgrounds. Okay, and HOMA is the de novo results. This is the KLF factor, which is good to see there, because if you remember, yeah, I know. here is also our KLF factor found in the random clustering. Adds NF-Y, NF-1 typical co-promoter factors. But again, high background versus high targets. And looking at the last one, I think we already looked at that. Yeah, no. Okay, so this is actually a good example. Similar to Google, it's like I don't really know much about this study, but just from looking at these mouse strains, they were treated, I believe, pneumoxia, hypoxia, given from the name. And we did not really find HIF or any specific factor that could be associated with that. Instead, we find at low percentages different factors that are known transcriptional activators like KLF, NF-Y, NF-1. So it could also be one result that this is because these are two different mice strains and they respond to the same signal. And so we will not find which factor they respond to by looking at the same response in two different genetic backgrounds, if that makes sense. Because if the same factor responds to it in both genetic backgrounds, it kind of nullifies itself in the enrichment, if that makes sense. What we're more looking at is where natural genetic variation is happening in the background of these mice. So now looking at that, I actually realized it might have been a little bit silly on my part to look at these pneumoxia conditions between two different mouse strains, and it probably would have been better to look at hypoxia versus pneumoxia, right? Because then it's a signal, and then look at within the strain. But these are just the conditions that I looked at. But nevertheless, I hope this was halfway useful. And indeed, I actually think sometimes it's best to see, an example of where things are not as crystal clear and where I believe there is no real motif that's associated with strain-specific gene expression, right? And I guess that makes sense. I don't know. But the key thing is, so these are the three different ways you can do your motif finding. Of course, there are many more, but these are the ones that I suggest you to check out. Okay, so I'm gonna share that video with you tomorrow because it's late and I have to go back to my Stormlight Archive book 4, which is amazing. You should definitely read that. Tomorrow I should also get the second lane from the sequencer, and hopefully we'll load the third one. So there will be a second wave of emails. And thank you for the patience for those that are eagerly waiting for the data. And unfortunately, have to enjoy their weekend instead of analyzing it. In that email with this video, I will also upload this Jupyter notebook. And so I hope someone can help you to set up the Jupyter notebook. I will also put in the link to the Jupyter notebook website. Quick comment here, there's Jupyter Notebook and Jupyter Lab. I recommend you to use Jupyter Lab just because it has a few more gadgets, and I think it's a little nicer than Jupyter Notebook. And what I would recommend you again is to just get Jupyter set up, download the packages, maybe do a quick Unix tutorial and Python tutorial if you've never done it. But then basically to just copy that notebooks, rename it, make sure that your files are named similar to what we did, and then just, copy, paste, go through it. And if there are any questions, feel free to reach out. If you want, I think I should also set up a Slack channel or something like that. Maybe we vote on that, because I feel many of you will run into the same problems, and it would be nice if we're all in the same channel and can just discuss that. But independent of that, always feel free to reach out to me. And with that, I wish you a good night and hear from you soon.